Through the introduction of a single nucleic acid mutation in a given sequence, this video will describe a number of point mutation examples. This is the given DNA coding strand sequence spelt out using alphabet spaghetti from my breakfast. When transcribed to mRNA, all the thymines will be replaced by uracil, and following this, translation will occur. As this is the normal non-mutant sequence, when translated, it produces a 5 amino acid long protein, beginning with methionine and ending in cysteine, as the 6th codon in the sequence codes for a stop codon. The rest of this video will be focused on explaining different types of point mutations using this sequence. A point mutation is a mutation that will result in the substitution of one base for another. This single nucleic acid point mutation, shown here in mRNA form, is a silent mutation. A silent mutation is when the single base change still codes for the same amino acid as previously. In this case, uracil has been mutated to cytosine. As both of these are pyrimidines, it is also an example of a transition. The nature of a silent mutation, and this example is no different, means that the protein's function is not altered, and the change will still result in the codon encoding for alanine in this case. The next point mutation introduced adenine in the place of cytosine. This is a missense mutation in which the change in base will change the amino acid, and therefore it may alter the protein's function, potentially even rendering the protein non-functional. It is, however, possible for a missense mutation to occur without altering the protein's function, in which case it will be referred to as neutral. The point mutation that has occurred here means that what previously was asparagine is now lysine. As this conversion changes a pyrimine to a purine, it is also an example of a transversion. The following mutation is a nonsense mutation, where the substitution of the base results in the codon now becoming a stop codon. This leads to an early truncation of the protein. In this example, the original cytosine is replaced by adenine, converting cysteine to a UGA stop codon. Again, this is a transversion as a pyridamine is substituted by a purine. Finally, this is an example of both a read-through point mutation and yet another transversion. Read-through mutations occur when the single nucleic acid change leads to a stop codon being converted to a codon for a specific amino acid. Thus, it results in a longer protein being produced. It is a transversion as a pyridamine has replaced a purine. This particular example involves the base change from uracil to guanine, resulting in glycine substituting the stop codon. Originally, the protein would have been truncated here, however, it now extends to the end as no stop codons follow. The last two amino acids are proline and glycine, bringing the protein up to 8 amino acids long, reaching the end of the given sequence.